Hi everyone, I am Saniya, and in this video, I want to show you how you can use R, mainly bioconductor packages, to extract information on rsIDs, which is SNP IDs. So if you're given a chromosome position and a location, and a specific build, which build it is, which I'll talk about, how can you go and extract the SNP ID, rsID information for that? So why is this important? Well, it's sort of exciting to me, and I'm sort of being a little bit of a nerd, but there was a new genome-wide association study that identified new risk loci. So these are SNPs, single nucleotide polymorphisms, for Alzheimer's disease. So it has like over 1.126563, so 1.1 million, over 1.1 million individuals. And this just came out about 3.5 weeks ago, which is awesome. So when I checked out what this data is, then I was given this information. So when I went and I downloaded it, so this is from Whiteman here, Douglas P. Whiteman, and so many authors, including 23andMe research team. So then I went here and I found that, Alzheimer's number 23andMe, the GWAS. So it has chromosome number. It has the position, which is GRCH37, tested allele, other allele Z, the p-value, which is this column, and then the N, the sample size. So it has all of that information, but it doesn't have the SNP ID. The SNP ID usually given by RS numbers, which is pretty important as well. So we can see the build, which is pos GRCH37 and this chromosome number. And there are a lot of SNPs here, which will be very exciting for us to analyze as well. So I think there are about 12,688,340 total rows here. So that's going to be a lot to sort of use other tools for. So if you don't already have it, if you have RStudio, then awesome. Otherwise, please install RStudio, and I'll have a video on the installation process for that. But if in R, if you don't have Bioconductor, which is the open source software for bioinformatics, which is awesome, then you can just install it here, especially version 3.13. So then what we're going to notice again is that we want to look for position GRCH37. So what you do is I'm going to need you to, as of now, to please just run this. If you don't already have BS Genome, then do BioC Manager. So once you've already finished installation of BioC Manager, which may take some time, once you already have this allows you to install bioconductor packages on your computer. And there are so many packages. I just love going through this. It's been so exciting for me to go through and check out all of the different packages. So you can just see packages and release. And there's just so many packages, out there. Analysis, statistical analysis, CRISPR, annotation for microarrays, analysis of translational activity. So there's so much stuff out here, which is really cool that we can focus on. So there's even one of mine, which is Cluster Profiler, which I love, a universal enrichment tool for interpreting multi-omics data. There's just so much out there, but we're going to focus on BS Genome. So once you install a BioC Manager, then you can just do install, and then you can just write the name of the package here, which is BS Genome here. So this is what you can run if you don't have it. So usually you just put the package name inside here. And then once you have it, you can just run library BS Genome, which is what this will bring here. I've already loaded it. So one thing you should do is to see all of the available snip locks and extra snip locks packages. You can just run this code available.snps, because at the time of this video, this is what we currently have, and this may change. So that's why I don't want to tell you to just use this database of SNPs. So again, we want to focus on GRCH37, because that's what the Alzheimer's disease data set has given us. But it might be important for you to just check and make sure that you know what the build is that you're looking at beforehand. So the other thing is that if you see, and you might also see GRCH 30. So you may see HT19 and GR37 could be the same as well. And you could also see HT38 as well. So you could just be familiar with those as well. These tend to be used interchangeably between these two. But in any case, what we're looking for is, GRCH37. So when we'll come here, we can see all of the available SNPs that there are here. And we're looking for GRCH37. So we want to look for the most recent database. So as of this video, it's dbSNP 144.GRCH37. So this is what I want to use. So I've already installed this package on my RStudio, but if otherwise, I can just directly install it like this here. And I can just install this package, this database here. And I can directly also look up on bioconductor.org packages release data annotation manuals. I can look up the same package right here, and which is what I have right here. So it was just updated recently, which is great. So it helps you access the SNPs that are stored in this database. So some of these functions were not working, which is also why I think it's helpful to create this video to help you guys out as well. But this has a database of all the rsIDs, which is super helpful because that's like the identifier as well for SNPs, and you might need it for different computations. So what you do is whatever package you end up choosing from this region over here, whichever package you end up choosing, you can just assign it SNPs to this package over here. That's all you do here. You just run this here, and what you'll see is when you finish running it, so I've already run it, but you'll see that you have a SNPs log objects for Homo sapiens DB human build 144, and reference genome is GRCH37.P13, and number of SNPs is a lot. So it has 135,276,726 SNPs that it has information on. So one of the things is that, there's a shortcut. If you just want to see that this works as well, so you can just run this command on chromosome 22, which is chromosome 22 SNPs by sequence name, SNPs. So you can just take this here and you can just move this here, and you can just see which SNPs are directly in chromosome 22 that it's found. So you assigned your SNPs just to this. You could have just put this in here, but it's in here. You could have just had something like this, but it's just too tedious. So we just kind of assign it to SNPs just to save time. And what you'll see is that this is chromosome 22, stitched G-post position with so many different SNPs here, which is about, so it's a small chromosome 22, because you can see the chromosome shrink in size, along the karyotype as well. So we have 23 pairs of chromosomes, and they usually tend to get smaller in size. As you can see here, they tend to get way smaller in size. So sometimes it makes sense to just see 1, and then 4, 5, 6, 7, 8, 9, and then you can see 22. So you can see it just really tiny compared to chromosome 1. So like looking at SNPs here is going to be much faster for this demo right now. So you can see it has the SNP, the chromosome number, it has the position. This is sequence names, is also just the chromosome number. This is the position on the chromosome. And what we really want, which takes the cake, is we really, really want is the rsID numbers. That's what we really want. And that's what we get. That's all we want, is we just want a mapping from the chromosome number and position to this rsID. That's what we want for this Alzheimer's disease data set here. We just want the chromosome number and the position to be mapped. So that's what this does. So you just give in the chromosome number that you want as a character. So if we just wanted for the chromosome 22, it'll give us all chromosome 22 and all the positions, whatever it has in this database. So instead, I wanted for all the chromosomes. I wanted for chromosomes 1 all the way to 22, because our data set starts at chromosome 1. And then as we scroll through, you can see chromosome 2, the leftmost column, you can see 8, all the chromosomes here that we have from this data set here. Sorry, from this data set here. So basically, that's what we want to get. So that's what this chromosomes vector is, and it's a sequence vector. So sequence basically, gives you a sequence of numbers, numeric values from 1 to 22, and as character just puts them in quotes. So this just becomes 1 all the way to 22. So what I did, it took me a long time to make sure you have a lot of space on your computer at the time, or you can try running this chromosome at a time. As well, you can just do 22. Then if you want to have like chromosome 3, for instance, you can just do chromosome 3 SNPs. It would just be like 3 here. You just put in a 3. But I want to get all SNPs information. So I'm putting in this character vector. You can put in a vector. If you want to get, for instance, chromosome 3 and 4 SNPs, then you could also just put this in here. You can also equivalently just do this and have a vector of C3, 4, and then you can also run those SNPs too. I really hope this works. I know I have like a lot of files open. Like you can see this is like 10.9 gigabytes. That's like a lot of information. That's what I'm saying. So So anyways, to get all SNPs info, we can just put in this giant character vector that essentially is all of this. We can just put this in here. Okay, so I think I got it. I think I just got really impatient. Okay, I'm a very impatient person. So you can see that this is 3 and 4 as well. And then this is like a position. And then this is also position here for chromosomes 3 and 4. This is what this is. It's like a GRanges object. And you have the SNPs, which is great. That's what we want. So we want to be able to have a database of all the SNPs information for all these 144 build for this GRCH37, which again, we want that because of the Alzheimer's disease data. So it really depends on whichever build you're using. We have that, and that's what they're going to map to. So I ran this, and this took some time. So so once I finished running this, so you can already see here that this is going to be unstitched with 5 slots here. So I can just show you what this looks like. I can just run all SNPs information, and it's got like pretty much all of them, 2 metadata columns. It has the position, the chromosome number, the position, and the reference SNP ID right here, which is great. So that's exactly what I want. And then what I want to do essentially is I just want to overlay like these chromosome numbers and positions. I just want, and these rsIDs, I want to kind of merge this information, chromosome number and position, with this chromosome number and position, so that I can get a column of the, I want a column essentially of these ref SNP ID. That's what I want. I want to have the ref SNP ID. I want to have this. I need to have this here. So I need to map them somehow. There are many ways you can go about this mapping process, but one of the ways I went about it is the following. So I read in this GWAS data set here. I read this in this text file in, I read red.delim here. So I'm reading in this file as GWAS data. So if I check this data out head of this, it's chromosome, position number, tested allele, other allele, Z, the p-value, and the n. So it's this nice little table. It's very cutely read in here. So this row corresponds to this row over here. It's all perfect, which is great. Then what I did is I used this function that's called make GRanges from DataFrame. So I put in the name of the file. So if you want, you're already pretty much done when you come here. And so if you just wanted to find some database, you can use that. But I'm just showing you an example of how to merge it and how to actually use this database to retrieve what you want. For instance, for me, it's extracting the rsIDs, the reference SNP IDs. See, I call them rsIDs from a GWAS data set. So then I can call this function that's called make GRanges from DataFrame. So it's going to take in this data frame, the data frame with the information. And what I'm going to want to do is I want to sort of set this so that it knows seek names field. So seek names field. So seek names is just the name of the field that has a chromosome number. So in this all SNPs info, we're trying to go from a data frame like this to a GRange object, something like this, because then there's code that we can run to do this overlapping very seamlessly, which is great. So what we want is we want to see this here and convert it from a data frame to a GRanges object, to a GRanges object, genomic ranges, essentially. So it's asking us, what is the field name? That's what field is essentially asking you. Hey, please give me information on whichever column heading in this data frame has the information that I can use to create this object for you. So I'm saying the seek names field, the field in this data frame that has the chromosome information is called CHR. And because it's a SNP, it's one nucleotide, it's one base position, just one, then the start field and the end field are essentially the same. It's position GRCH37 and position GRCH37 is the position. It's just the starting and ending position. And since it's SNPs, then GRanges will automatically understand that since the start field and end field are just the same, it's just one spot in the DNA. That's what a SNP is, a single nucleotide polymorphism. And DNA is made up of polymorphisms. And it's also made up of single nucleotides, A, P, G, C, that are stitched together to form that double helix and a sugar phosphate backbone. So that's all you put here. You have to put the start field and end field here. Keep extra columns, I said is true, because we would want to also have like tested allele, other allele, z-score, p-value, and we just want it all to come for a ride. So I create this here, and we can just see, I can also just call this directly, and we can see that this has all the SNPs here that we wanted. So if we go to the bottom, what we see is that we have, as I was pointing before, let's go right to the bottom. We have about a 1, 2, 6, 8, 8, 3, 3, 9, because one of them is a heading. 3, 3, 9, And that is exactly what we observe here. You know, we have the 339,12688, so 12,688,339. Personal complaint is sometimes I wish these languages just automatically had the, you know, the commas after every three. So I know in India, they use like lakh and rupees and different commas after digits, which confuses me a lot. Anyways, so now we have these two objects. We have this all SNPs info, and then we also have this genomic ranges GWAS SNP. So we have these two. And what we want to do is we want to just find the overlap. So we have the chromosome number and we have the position here. And we just want to kind of combine these together here. So you can see that it has the other stuff here. It's got the other stuff, like it's got the tested allele, other allele, Z, P, N, and this is kind of like extra information that you're getting. And similarly, this is also extra information, which is actually what we want. You know, it's the other additional information that it's carrying, which we got by saying that we wanted to keep the extra columns for this part gave us essentially this. So then this next part is something you just have to kind of make sure you keep the logic there. So you can have Q hits. So you want to find the overlaps and see if the indices that overlap between the two. So you want to just kind of do like an inner join, just find whichever SNPs, so this is the bigger one. This is the database of all the SNPs and information from this build. And this is the SNPs in the Alzheimer's disease data set that we want to figure out. So we want to just basically find overlaps between them. So basically find out which ones are in here, and actually all of them are in here, but like we want to just find for these SNPs and locations, what are their reference SNP IDs? We want to search through this database of 130 million, over 130 million different SNPs, and sort of get the rsIDs for these. We want to basically find the mapping and move this down here. We want to just move this here. We want to be able to map it. So to do that, all you have to do is run this Q hits and subject hits. So all you have to do is what you'll notice is that they all are pretty much the same here. So this part right here, find overlaps, query this is the same. And this is also the same here. The only difference is this part and this part. Query hits, subject hits. Subject hits becomes sub hits, and query hits becomes Q hits. So this other part is exactly the same. And while you register that, I'm just going to so this is exactly the same. That's all you have to change, is you just have to change this. That's all that changes there. And so you can see that query hits and subject hits are what differs. Then what you have to do is you have to take your GWAS data. So whatever came first, and so the thing you have to do is you have to see, okay, query is this one here. So I'm going to put this data frame here. Whichever I put here in the query, I'm going to put under query hits. So I take the data frame for GWAS SNPs. So now I want to make a data frame. So I already had read in GWAS SNPs came from this data frame, GWAS data. So since this was my query, I'm putting this first and I'm using Q hits for it. So essentially, whichever came first, that's the query. And then that's what you're going to put in query hits. So in other terms, just kind of like, so you can understand that, like, I want to explain that. So you have the Q hits here, and then this Q hits kind of comes from this GR snips. And then this GR of our snips here has this GWAS data here. And then this GWAS data will then come here. That's sort of how it ties in together. And then for the subject hits, because again, Q hits is for the query. That's the query part here that we'll be using. So that's how we have those connected here. Query, Q hits. It tells you the rows to use for GRWAS snips. And that's what this tells you. And then you use the corresponding data that gave you GWAS snips, which is GWAS data. That's a data frame, you know. So if we look up str GWAS data, it's going to be a data frame. So we can move that here. And query hits, Q hits this is for so I'm just going to maybe mention this below. So Q hits gives us query and sub hits gives us subject data frame rows to use, which here is all snips info df. So these are the overlap. The rows that were found between these two. And all of these, hopefully, GOS snips should be found in the all snips data frame. So they should all be found there. So this gives us a subject data frame to use. So the same idea kind of goes for this all snips data frame, which is the subject. So and this all snips data frame is currently, if you look up the structure of it, it's like a genomic ranges, GRanges object, but you can just directly turn it into a data frame like this by just doing data.frame and then this. And if not, you can also just directly kind of, you know, put that in here like this too. It doesn't matter either way. You can have it like this. So in a data frame, again, you have the rows, comma, columns. So if you don't have anything here, you just take all the columns. If there's no argument here, if there's no like C1, 2 or something, there's nothing like that. You just take all the columns and all the columns. And this is just the rows. So you're selecting specific rows. So you can just take this one and just take the subhits. Subhits just tells you which rows to take from all snips info. And the Q hits tells you which rows to take from GWAS snips. So then what I get after I do that is I essentially get this as mapped. That's awesome. And I kind of want to just show you like how that looks. So we have one and this here, which is from the first data frame, which is exactly mapped to this one. That is awesome. So what we got is we got this part here, which is coming from, this part is our GWAS data. And this next part here, which is great, is just SNP database. So that is awesome, because now we have this data frame here that we can write out. So we have the chromosome and the position, and then we also have they map. So 1 and 14,860 is the same thing here. And then we have the SNP ID. So we have all the SNP IDs that we want. So I can also just directly view overlaps. Going to take some time, so I'm not sure if that was a good decision. So that's just, in other words, that's just how you can see all of these, you know, chromosome positions that you want. We can maybe, instead of viewing this, let's just also view rows 1 to 50. Like just see the first 50 rows. Okay, that's taking a while, but essentially you can just write out the CSV when you're done as overlaps and then the file path that you want. So I just like CSVs more than text files, which is what it had come in. So I can just call it map snip. So this is the snip data file that I can use. So great, now we can just see the reference SNP ID, alleles as ambiguous, and then and we have all of these for the first 50 or so as well. We have all of this stuff here. So it also has these numbers as just like the overlap numbers in the end that we ended up using. And what you can just see again is that we got what we wanted. We have the chromosome number, position information, allele information, Z, P, and sequence names. Again, this is overlap redundant. You can just eliminate those two if you don't want them. Position, strand, reference, SNP ID as well. You can have all this, and I just kept them for now just for sanity checking, so we can just see that these two are the same, and these are also the same as well. And we have the SNP ID, which is all we wanted. And we can just see we have that for all of these, kind of scrolling down arbitrarily as well here, you know, 21st chromosome position. You know, that we're just checking that this is the same here. This is also the same here. And we've got the SNP information, which is great. So that's just an example of how you can do that. So I just hope that you found this helpful and that this kind of shows you how you can do the mapping, but please make sure you have a lot of space that you would need in you know, your RStudio, and you've closed unnecessary files on your computer so that you can do this seamlessly. So I hope that this was helpful. If you like this, please subscribe to my channel, but just trying to show you some helpful tips for kind of mapping, because there are other tools out there again that I kind of talked about. If you're interested in them, there's this other tool. I have another video on this where you can also find out the SNP ID as well. You have the build HG19, CH37, so you can find out the reference positions and informations from this website. And I show an example in another video, but what I was showing you is how you can do this in R, and not just for 100,000. You can do this for many, many SNPs at a time. So yeah, again, just a quick recap again is that you just have to kind of just check all available SNPs that there are, all packages, figure out if you're looking at this one, maybe you might want to use 151, the most recent build. Then you want to also figure out, you know, assign that database to SNPs. then then use all the chromosomes that you want to use and have all SNPs info here, and you'll have your database of all the SNPs that you need, and then you can just map it. So yeah, please let me know if you have any questions at all. So I hope you guys have a really awesome day, and that this helps you guys with your research as it has helped me with mine so far. Thank you guys.